Okay, welcome. We're going to start a discussion. This is lecture 4 on genetics. Now, as we move through this, we're going to have some terms that I want you to be familiarized with. First off, when I talk about the genome, that is the entire collection of genetic material of a cell or virus. Traits are heritable. They're passed on from one generation to the next. Gene is a unit of genetic material, coded usually on the DNA, for a particular trait. A genotype is the genetic makeup of an organism, whereas a phenotype is the observable physical traits. Nucleic acids that contain genetic information. For the eukaryotes, it is DNA. For RNA or DNA, it's usually viruses. So usually you're going to find prokaryotes and eukaryotes having DNA, but their genomes are going to differ in size and organization. And I'll show you a chart to bring that to your attention. The genomes are organized in chromosomes. So if we look at the prokaryotic genome, complexity, it's relatively simple. It's just a chromosomal DNA. Sometimes you get these smaller loops of DNA called plasmids. A lot of attention has been played on plasmids because of the fact that they can be transferred from one organism to the other and enhancing some of the pathogenicity or virulence of this new organism receiving this plasmid. For example, antibiotic resistance. Some of those genes will appear on plasmids and thereby can be transferred from one related organism to the other. When we talk about eukaryotic chromosomes or eukaryotic genomes, the chromosomal DNA for eukaryotic genomes is going to be in a linear fashion. And it's going to be held together, looped together, or condensed together as a chromosome. Now, there's a difference though, because with many eukaryotes you have the mitochondria and the chloroplast, and their DNA is very similar to bacteria. Also, you will have some eukaryotic organisms that have plasmids as well. These are the smaller loops of DNA that carry certain genes for certain traits. The chromosome for prokaryotes, they're few, generally only one. Usually it's circular. Eukaryotes, the nuclear chromosomes are linear, and you may have many. For example, humans have 46 chromosomes, which really is 23 pairs. The last pair being the one that defines whether one is male or female. The location of the chromosomes are in the nucleoid region for prokaryotes, but the nucleus for the eukaryotes. DNA is organized by, in prokaryotes, a histone-like protein. For eukaryotes, they are histones. Okay. More on terminology, when we talk about a eukaryotic chromosome, many of them are linear wound-up type with the use of histones. These proteins will act as a spool for wrapping DNA around. Plasmids, as I mentioned, are located in both eukaryotes and prokaryotes. They're mostly just circular loops, and they will confer survival advantages to the cell. As I also mentioned earlier, keep in mind that mitochondria and chloroplasts have a small circular chromosome. How small? Well, the mitochondria on average is about 16,000 base pairs. Now, here we have the virus. Now, the virus, you have to be aware, can either be DNA or RNA. Okay? We'll get more into this when we get into viruses later on. Here we have the chromosomal DNA for a bacteria, in this case a prokaryote that we've talked about, and it's kind of a loop that's kind of squiggled around here. You may get some smaller loops of plasmid DNA here. You can see this also in eukaryotic genomes. You will see, again, linear arrangements of chromosomal DNA for the eukaryote, but you got to keep in mind that The mitochondrial DNA and the chloroplasts will have small loops. Again, they're very similar to bacteria in their genomes. When I talk about nucleic acids, DNA and RNA govern the cell life. The nucleic acids consist of phosphate, a sugar, which is either deoxyribose or ribose, and a nitrogen base, which can be either adenine, cytosine, thymine, guanine, or, and it's very unique, only be found in RNA, uracil. You'll review the table there, and the structure is unique. And that DNA structure is a double helix set in anti-parallel arrangement. So in other words, one goes in one direction, one goes in the opposite direction. You always have pairings of adenines with thymines and cytosines with guanines. That is because where they bind together are held together by hydrogen bonds. And this is in the nitrogen base area. The sugars and phosphates are held together by phosphodiesterase bonds. Here we have the arrangement here. The nitrogen bases, adenine, guanine, cytosine, thymine, uracil. Adenine and guanine are in the purine family, whereas cytosine, thymine, uracil are in the pyrimidine family. And you will find that adenine, guanine, cytosine, and thymine are found only in DNA. Adenine, cytosine, and guanine and uracil are only found in RNA. So there is somewhat of an overlap of the use of purines and pyrimidines and somewhat unique cases as well. Here is our designation. This is a phosphate group. Here is the sugar. Now, if you notice right over here at the 2' off of that carbon, if it's ribose, it would be an OH group, a hydroxyl group. If it's deoxyribose, then there would be just a hydrogen. Then we have the nitrogen bases here, and you can see the designation of the purines versus the pyrimidines. And this is what we have here. We have nitrogen base pairing occurring, and that creates the rungs for the double helix structure. A's with T's, they share 2 hydrogen bonds between them. C's with G's, 3. That's why you can't really have easily or stably a mispairing of, let's say, A's with G's or T's with C's. DNA orientation. This is anti-parallel, which means that each strand is oriented in a 5', 3' direction. Each strand is in the opposite direction to the other. Anti-parallel directionality allows the complementary base pairing of DNA to properly associate. Now in RNA, the ribonucleotides can have some hydrogen bonding, but remember that RNA is single-stranded, but it can have some bonding with sections of the same molecule as long as it comes in contact and with the A's with the U's now. Okay, A's will bind with U's in RNA, C's will of course bind with G's. You can form these unique loops and helical folding within itself. Again, phosphate sugar base, and you can see here's the DNA, and notice that you have the phosphodiesterase bonding here between the phospho group and the sugar group. Now, if you look at things from and this is a diagram that's set up by the author, obviously, with one side of the paperclip being a 3', the other 5', as you attach and continue to build on these from the phosphate grouping, 5' to the OH here, 3', you have to have sort of this anti-parallel arrangement. They complement each other here, but they are in different orientations, 5' to 3' here, the opposite 5' to 3' here. Now there is what is referred to as the biological dogma, the dogma of, the central dogma of biology as it's called, and that is general flow of genetic information goes from DNA to RNA to proteins. Now, to make DNA to RNA transcription, That is the process to help you to understand that, and it can get easily confused. We are transcribing. Transcribing means that you're making a copy. So you're making a copy from DNA to another copy in RNA. So you're making a copy from one nucleic acid to another nucleic acid. RNA, to make a protein, involves the term translation. So you are translating the genetic information from a nucleic acid, RNA, to a peptide strand made up of amino acids, and that's the protein. One other point. Reverse translation can occur via reverse transcriptase enzyme. Now, the reverse transcriptase enzyme takes RNA and makes a DNA copy from it. Now, this is also something you have to be aware of. The DNA can copy itself. This is referred to as DNA replication. So we have DNA. We have to separate the two strands so that we can read off one of the strands and make a copy of the genetic information in the form of RNA. That's transcription. This information will then be sent outside into the cytoplasm, read by the ribosome, which will translate this information into a proper sequence of amino acids here, which make up the protein. This RNA can also be reversibly transcribed into a strand of DNA. You need what is called reverse transcriptase as the enzyme to do this function. We are going to focus first on DNA replication, which is the process by which the cell copies its genome before it divides. Now, usually this requires a DNA unwinding. As I mentioned, you have to split the two strands, the double helix. So unwinding the original DNA copy, then you copy it. Then you are rewinding the parent DNA and the newly made DNA. Really what goes on is what is called a semi-conservative process, where one strand of the original DNA acts as the template to make the complementary strand. And those two strands, you've got one old, one new. On the other side, with the other original parent strand, you will have a template which acts as a template. You will make a second strand here, and what you end up with is two strands, half containing the old information, half containing the new information, based on the old information as a template. And this is referred to as the semi-conservative method of replication. Now, the size of DNA differs in various organisms. For example, humans have a DNA genome of 3 billion bases. E. coli, though, is only about 4 million bases. We have to talk about a couple of terms here. Mutations, these are changes to the DNA code. Now, you have to think about this. Replication, though very fast, is also accurate. There's a low error rate, a low mutation rate. Enzymes are proteins that facilitate chemical reactions in a cell. So we're going to talk about a couple of enzymes that play a key role in this replication process. But at the same time, as you're making these copies, you have an error correction rate. And the error correction mechanism that's involved with this may be part and parcel of the actual enzyme, in this case, for example, DNA polymerase. Okay? Think about it. We've said that E. coli replicates every 20 minutes. So that means within that 20 minutes, besides the cell growing a little bit, building up some of its organelles, having metabolic activity, etc., it will also have to copy its DNA genome of about 4 million bases. Now, to start replication, prokaryotes start with a collection of protein factors called a primosome that collects at the specific point on the chromosome called the origin of replication. A primosome includes enzymes, helicase, which unwinds the DNA, primate, which lays down an RNA primer to jumpstart the replication process. Single-stranded DNA exists and acts as a template to synthesize the complementary DNA strand. Newly made strands, half old, half new, as I mentioned, are called daughter strands. Again, this is what I was referring to earlier with the semi-conservative method. So as we separate the two strands that we're going to make copies of, this is where we get into what we call a replication fork, the exact point where unwinding of the DNA occurs. And due to the bidirectional progression of DNA replication, there are two replication forks that form. This process continues until you have two separate daughter loops of DNA are formed. Now in the process, you've got some serious enzymes that have to play key roles. Helicase breaks down hydrogen bonds between the nitrogen bases. Single-strand DNA-binding proteins will bind to the DNA to maintain DNA stability as a single strand. So in other words, once we separate them, they can easily re-anneal together, but instead you have these proteins that keep things separate while the replication process occurs. Two other important enzymes, gyrase and topoisomerase, relieve the strand coiling tension as the helix unwinds. Then we have that other enzyme, PRIMASE, which adds RNA primers at the start of the replication site. Heads up. Gyrase, topoisomerase, helicase, these are all targets for antibiotics, which will block the DNA replication and hence lead to the death of the cell. Here's our bacterial chromosome. Here we have two replication forks, one on this side going in this direction, one on this side going in this direction. Enzymes will separate the two. The special proteins will keep this open and separate. You will have another enzyme that will read off the parent strand and start making a complementary copy. So wherever there is on the parent strand an A, there will be a T on the new daughter strand. Wherever there's a G, there will be a C, and so forth, until you've made two completely new strands here. Because what you're going to end up with is half old, half new. Now, this is very important. DNA polymerase III. This is the main enzyme to build DNA and uses the RNA primers to start and then proceeds in a 5', 3' direction for DNA replication. You see, primers are needed for polymerase III, because it can't just start from scratch. Now, there is another DNA polymerase called DNA polymerase I that removes RNA primers, replaces the section with a DNA strand. So in other words, after you've had the primers, you remove them, you fill in that little gap. There is a enzyme called ligase. It acts like glue. It's the enzyme that glues junctions of the sugar phosphate gaps together after the primer replacement or between gaps in OKAZAKI fragments. Now, due to anti-parallel arrangement, there will be one leading strand where the DNA molecule is copied in the same direction as the helix is unwinding, and one lagging strand. This is the complementary strand copied in the opposite direction of the unwinding. As a result, you're going to have this start, stop, and what will happen is you'll have these very small gaps here between these smaller strands, and this is what is referred to as OKAZAKI fragments. So how do I fill in to make this nice and solid? You use DNA ligase that will go through and fill in the gaps. This is going in a 5', 3' direction, the same direction as the helicase, but this one, because it's kept out here and because it can't do 5', 3' at one point and just keep going down, it goes for a certain distance, stops, pops off. Because once it bumps up to again another area of DNA double strands, it stops there. Here are the single strand binding proteins that keep this double helix stabilized. Here is the helicase, which is right here. Okay. Now, key enzymes in the DNA replication, I've already mentioned most of them here. I encourage you to review this chart as you go over this particular chapter. Replication direction on leading strand is continuous. There's no problem with that. But here's the problem. You can only go 5', 3' for a certain distance and stop. Meanwhile, behind you, you're still forming single-stranded. So what happens is you're going to have the DNA polymerase jump in where there's new strands, and it will go forward for only a short distance until it bumps up against double-stranded DNA. This is where we get into the replication direction on lagging strands. This process is discontinuous. It's kind of start, stop, start, stop, start, stop. It results in OKAZAKI fragments. And at the end of replication, the two separate DNA helices are formed. And I just mentioned this earlier. This is called the semiconservative process. One strand of the double helix is old, one's new. Prokaryotes and eukaryotes replicate DNA with differences due to the size and length of the chromosome, linear versus circular structure, packaging requirements for the eukaryotic DNA, and the requirement in eukaryotes for more protein factors. Cell division time, as I mentioned to you, you can take an example using E. coli, 20 minutes. Eukaryotic cells, 18 to 24 hours. So as you can see, one, there's a lot more DNA being replicated with eukaryotic cells, but on top of that, the species that is bacteria has to move very quickly and has to have very little error rate. Now, why I say little error rate? Because errors cause mutations and that can lead to a reduced fitness in some cases for the survival of that species. Now we're going to focus in on protein synthesis. This entails transcription and translation. To help you with the clarity, I've mentioned this before. We transcribe, make a copy of the genetic information from one nucleic acid, DNA, to another nucleic acid, RNA. We translate the genetic information from nucleic acid sequence, RNA, to an amino acid sequence, protein. Now, protein synthesis, otherwise known as gene expression, you read the genetic information, use it to create gene products, in this case, proteins. The key enzyme here is RNA polymerase, which performs the translation process. In the translation process, it consists of 3 steps. Initiation, that's where the RNA polymerase binds to the promoter, beginning the region of the DNA, the gene in the DNA. The RNA polymerase unwinds the DNA to expose a single-strand template of the gene to copy into the RNA. Then we go through elongation. The RNA polymerase advances along the single-stranded DNA, and it's inserting complementary ribonucleotides. If there was an A on the DNA, there would be a U in the RNA. If there was a C on the DNA, there would be a G in the RNA. If there was a G in the DNA, there would be a C in the RNA. And if there was a T in the DNA, that will end up with an A in the RNA. Finally, after all of this work, we come to termination. There is a termination signal on DNA and in DNA that signals the RNA polymerase to detach from the DNA, and the newly made RNA transcript is released. Translation occurs in the nucleus for eukaryotes, whereas translation occurs in the cytoplasm for prokaryotes. If you take a look here, here's our transcription process. You notice that you start off with DNA and you're translating the information into a single strand of RNA. We've talked about the steps: initiation, elongation, and termination. If you're still somewhat fuzzy about this, take time to review this chart. Notice the key player here, RNA polymerase, which will unzip, read the information off the DNA to make the RNA strand. Sorry, had a little thought there. Okay, moving on. Now keep in mind also, I mentioned to you before, reverse transcriptase. This is an enzyme that uses RNA as a template to make a copy of DNA. We would call this copy DNA cDNA. Some people think it's copy DNA. It means complementary DNA, because what it does is it removes all of the information from the actual original genome. And so all you get is what purely codes for that particular gene product. Now, certain viruses, particularly retroviruses such as HIV and things like that, and certain human cells make reverse transcriptase enzymes. Now, there are three types of RNA. Messenger RNA, symbolized by the small case m RNA. These are linear RNA molecules that carry genetic information stored in the DNA. They use a triplet of RNA codons. This is referred to as RNA nucleotides, excuse me, and this is referred to as a codon, to code for each amino acid in the sequence. Transfer RNA, and it starts off with a T and then RNA, is a cloverleaf-shaped RNA molecule, much smaller, that brings the correct amino acid to the ribosome for protein assembly. A section on the tRNA is called the anticodon loop. It is complementary to the messenger RNA codon. Ribosomal RNA has a small case r before the RNA. They have a three-dimensional shape and proteins attached to this RNA framework to form ribosomes. So in essence, what a ribosome is consisting of ribosomal RNA and a cluster of different proteins that attach to the ribosomal RNA, almost like you're adding walls and concrete and all this other stuff to a steel beam skeleton or a structure, okay, a framework. Now, transcription makes all types of RNA in the cell. Here's one interesting point, though, with eukaryotes. When you've made your RNA, this is all occurring inside the nucleus. The RNA doesn't completely leave immediately, but it may get trimmed or altered slightly. Prior to messenger RNA leaving the nucleus, certain segments of the mRNA are clipped out and the remaining fragments are fused together. The fragments that are retained are called exons, and they are spliced together, and they will still code for proteins. The fragments of the messenger RNA that are clipped out during the splicing process are referred to as introns. And there is a complex located in the nucleus that performs the mRNA splicing. It is called a spliceosome. Here's my messenger RNA. Here is my codon. Here is my transfer RNA. Here is on the structure the anticodon. So this will bind in complementary fashion with this codon here. And then finally, the ribosomal RNA, rRNA, and it takes a very complex stem and loop structure so that proteins can bind to it and make the final kind of roundish or globular sort of structure. Now, the genetic code and amino acids. There are 20 standard amino acids and 2 non-standard amino acids. Selenocysteine and pyrrolysine are what they refer to as 2 non-standard. These are all coded from the messenger RNA, and they must be arranged properly. In reviewing the table that you'll see, 5.4, each amino acid is coded by at least 1 codon. But here's the deal. Think about it this way. When they started to, in the late 50s and early 60s, say, well, okay, we know we've got these 4 bases. How do we eventually formulate them to code for the 20 different, or later on with the 2 non-standard amino acids? How do we do this? Well, if you take 4 single base pairs that are different, you only get 4. If you take doublets, in other words, 2 of the nucleotides together, you get 8 possible combinations. No, that's not enough. If you went to 3, You have more than enough to equal the 20 amino acids, and as well as a start signal and a stop signal. And we'll see that change in a second. Many of the amino acids have actually more than one codon which will code for them. This will allow for mutations and the third nucleotide wobble with the same retention of the amino acid sequence for the gene product. Now, this may look complex, but let me just help you here. On this particular side here, the left side, this codes only for the first base. So these choices here are just for U's. These choices are just for C's. These choices are just for A's. So in this entire column here, you will just have the first base being A's, here, here, and here. Then what you want to do is look at the second base. Well, the second base, all of those with the second base in this column here have a uracil. In this column here have cytosine. In this column here, they have adenine. Finally, we get into the third base. This is where they get the UCAG. The third base would be G here, would be A here, would be C here, and would be U here. And consequently, do the same thing throughout here. Now, what you have to understand is this, and this has done some serious experimental work. This codon and this codon both code for the amino acid phenylalanine. Now, this one, and yes, there's more. 1, 2, 3, 4. All 6 of these code for leucine. Now, if you take a look over here, these 4 code for alanine. Okay, serine is coded for these 4 here, as well as these 2 over here. So you have some cases where you have not merely 2, but up to 6. In the meantime, you have 1, 2, 3 that code for the stop signal. Finally, how do we start all of this? Well, there's usually an AUG that always starts the codons. It will code for methionine. It is also the code to start. The methionine, if the cell doesn't need that at the start of the protein, eventually that will get clipped off later on. So here we have the genetics and a comparison between the prokaryotic and the eukaryotic cell, where you have transcription and translation. In a eukaryote, the transcription is in the nucleus, the translation is in the cytoplasm. In the prokaryote, both in translation and transcription occur in the cytoplasm. Now, eukaryotic cells must process their mRNAs before translation. We talked about this example. We will remove introns and splice together the exons that we save. So now you have one whole unit composed of exon 1, 2, 3, but a much shorter messenger RNA. Translation. This is the process to assemble a protein. Now, each ribosome must first attach a messenger RNA to the ribosomal subunit to become a functional ribosome. The ribosome has 3 binding sites within it. It has an A site, a P site, and an E site. The A site is the acceptor site. The incoming transverse RNA enters the site. The P site is the peptidyl site. That's where transfer RNA with the growing peptide chain shifts into this site. E site, the transfer RNA encounters a stop codon. Termination factor enters this site to trigger the end of the process. Now, the process itself consists of 3 steps, similar to what you may have heard before. Initiation, elongation, and termination. Initiation, the ribosome attaches to the messenger RNA, scans for the start codon, and adds the first amino acid. The tRNA, which codes for both start and methionine anticodon, binds with the messenger RNA codon. The elongation step, and we got to understand this could be a very, very long or very, very short protein. The ribosome will elongate the protein while reading the messenger RNA, and the tRNAs will shuttle amino acids to the ribosome. Okay. Termination occurs. This requires a functional stop codon, or what has been referred to as a nonsense codon, that signals termination of the process in the disassembly of the ribosomal complex. Now, a polysome is something, you don't see it as much. You see it a little bit with eukaryotes, but you see it much, much more amply with prokaryotes, because in bacteria, because they have such a short lifespan, the messenger RNA will have multiple ribosomes attached onto that messenger, and they will be reading off a single messenger RNA at the same time. So they have the rapid production of a lot of that protein that's needed. Now, post-translational modifications, these are situations where the protein is altered either in size or may have various organic or inorganic factors added. Here we have translation, and you can see the E, P, and the A sites, the anticodon, which complementary binds to the messenger RNA, and of course, on the end of the tRNA, the amino acid. These will be added on one at a time, and as you can see here, eventually, when you get to complete termination, the termination factor comes in here, and then you have everything disassembled. So large subunit, small subunit come apart. The last transfer RNA will unleash that chain of amino acids. So everything kind of pops apart. Here is the formation of a polysome. And you can see this by a transmission electron micrograph showing simultaneous transcription and translation. So as you're transcribing, these ribosomes are going to jump on and do the translation. Okay? In controlling the protein synthesis, it is essential that we do this for all cells. In short, Mother Nature hates when we have excess proteins made that we have no need for. At the same time, we have to have a signaling mechanism to start the process if we need that protein. But there are processes that occur in both prokaryotes and eukaryotes. One of them is pre-transcriptional regulation. You regulate how the messenger RNA is made for a given gene. You have structures that act as switches turning on or off, dependent on the needs. They're called operons. You have quorum sensing. You have epigenic control. This is where you get the DNA being methylated at its nucleotides, and so it stops DNA replication or DNA, the process of basically transcribing. You also have the recruitment of transcription factors. When we talk about post-transcriptional regulation, this is after we've done transcription. So now we have to have a control of gene expression by regulating how often the messenger RNA is translated into proteins. Think about it this way. If the messenger RNA is there, more often than not, you're going to have it ribosome bind down and read it, ribosome bind down and read it. But if you have a shortened half-life or a lifespan that gets reduced of the messenger RNA, there you have a means of doing some regulation as well. So how do we have this post-transcriptional regulation? You recruit ribosomes to bind to the RNA. Okay, that's nice. That's an upward sense. Controlling mRNA stability, small non-coding RNAs that may block the reading of the messenger RNA, riboswitches, which are small amounts of RNA that will act as switches or blocking agents. And finally, and you only see this in eukaryotes, you control the RNA processing and the nuclear export of them. Because remember that the RNA has to leave the nucleus so that you can do the translation process, which occurs out in the cytoplasm. Here we have transcription, here we have translation. Here we have the pre-transcriptional regulation examples, as well as the post-transcriptional regulation examples. The last one, eukaryotes can also control RNA processing and nuclear transport. There is no nucleus in prokaryotes, and therefore you can't use this process in them. Now, pre-transcription regulation. This helps the cells control when and how often transcription occurs. This regulates the production of messenger RNA. Operons are a collection of genes controlled by a shared regulatory event. They're found in select eukaryotes and prokaryotes. The elements that make up this operon consist of the following. Okay. A promoter. This is a site on the DNA where the RNA polymerase binds to. Two or more genes with a commonly shared task. Repressor, that is a structure, usually a protein, that blocks the transcription. And then operator. This is the part of the operon that the repressor binds to in order to block transcription. So there are two types of repressors here I want you to be aware of. The operon is off normally. Lactose is absent, glucose is present, so I don't need to make these lactose-utilizing genes. So how do I get the signal to start acting on this? Allolactose is not present, so the repressor remains active. The repressor is going to bind down into an area, and the RNA polymerase is not going to be able to get around this repressor. So it's as if you had flipped a switch, moved a boulder on top of the railroad tracks, train can't go. End of discussion. And downstream, you have to look at what is it blocking. Actually, in some of the examples, it's three separate but very important lactose-utilizing genes for metabolism. But let's look at another operon where the lactose is present, the glucose is absent, and the repressor is still acting presently. An inducer allolactose is made when the lactose is present, when the glucose is absent. Now, what does this allolactose do? It binds to, as you can see here, the repressor, which removes itself from the strand of messenger RNA. RNA polymerase will be able to ride down this DNA track to make messenger RNA. Okay? So here we have a system that's normally on. Here we have a system that's normally off. And it's just to get the right key for the right protein product to act and remove the repressor. Now, as I mentioned to you before, you have the different types of operons. The inducible, these are off by default. The lac operon is one example. 3 genes to help the cell in lactose digestion downstream. The operon is induced, actively transcribed, once lactose is present and the glucose is absent. Both conditions have to be present. Repressible operons, this is where they're on by default. They allow cells to halt production of substances that are already abundant. Arginine acts or activates the repressor only if arginine is present in sufficient quantities. When arginine supplies are low, the repressor loses arginine, becomes unbound to the operator and allows transcription to resume. Here you see this here. Cell is low on arginine, the arginine comes up, but you have an inactive repressor. It's not here anymore. So now what you have is a nice long situation where you can see the arginine building genes transcribed. The RNA polymerase will bind to its promoter and then buzz right past the operator. This is if the operon is on. If the operon is off and the cell has plenty of arginine, the abundance of arginine is going to influence the repressor so that it binds to the operator. And the cells here can have a field day also, to a degree, with the RNA polymerase there. The RNA polymerase can't transcribe the structural genes, so proteins to build arginine are not made. Okay. Next we have to deal with is a concept called the epigenome. This is getting a lot of attention, started in the late 1990s and really began to take off. And it's a collection of all chemical changes to the genome. Epigenetic regulation is a way to control protein synthesis. Yet you're not directly altering the appearance of DNA without changing its sequence. Okay, so this is where what happens is this. You have something called DNA methylation. This is only one version of epigenetic regulation. And this methylation process is used by both eukaryotes and prokaryotes. It's a process that uses adding methyl groups to DNA to basically silence the gene and prevent transcription. Methyl groups are usually added to cytosines. Okay, and you have other ones. You have acetyl groups, etc., that can be added on. The main point, though, is you're not damaging the DNA. You can reverse this process when you need to, but on the interim, you can shut down at the DNA level. Now, we have another process called quorum sensing. This is the collective sensing and responding to changes with a bacterial community to enhance survival. Coordinated protein synthesis through quorum sensing is the foundation for forming biofilms. All the bacteria get together, they basically start sensing their presence and start building a biofilm that enhances the survival of the entire amount of bacteria present. When we talk about mutation, this is a change in the genetic material of the cell or virus. But mutations are essential to evolution and variation within the species. Now, I want to introduce you to something. If you think about a strand, picture it in your mind right now. You have a strand of DNA, and you change only one nucleotide, just one, in that entire strand. This is referred to as a point mutation. So this change, this mutation, occurs with just one nucleotide in the nucleic acid. As a result, you can have any of the following. A substitution mutation occurs when the incorrect nucleotide is added. This can occur during replication or repairing of DNA. Insertion mutations occur when the cell adds one or more nucleotides to its genome sequence. Deletion mutation is opposite, when the cells remove one or more nucleotide from its general sequence. So what are the possible effects? Now think about this. If I change one, if you notice that some of those codons earlier in the RNA listing, hey, they were saying you could have 6 different versions of one particular amino acid. Well, what if I had a mutation and all I did was change the 3rd nucleotide of the codon? That means you have a silent mutation. The mutation does not change the amino acid sequence of the protein. You had a slight change, but what was then serine is still serine. A missense mutation. This is where a new codon encodes the wrong amino acid. You started off with serine, now you end up with lysine. Oops! Nonsense mutation. This occurs if the change leads to a stop signal instead of an amino acid. This leads to truncation, which think of it as like a cutting off, cutting short of the peptide will occur. So instead of saying, arginine, arginine, glycine, glycine, leucine, you have arginine, arginine, and then nothing. Because what you made was that change that turned into one of the three stop signals. We also have the concept called reversion mutation or back mutation. Yep, it can happen. It's a second mutation that corrects the original sequence that was changed due to a primary mutation. So let's take a look at this very quickly. Here's the original DNA. The base changes in the DNA template, the resulting codons in the messenger RNA, and it's translated as what you can see there. Maybe it's silent, so it's still lysine. Maybe it is a major change. This is threonine. Maybe it's nothing. Okay? Here we have the effect of an insertion. Now remember, an insertion is a little different than a point mutation because an insertion, you already have your set number of DNA, you just add a letter. Or you can add, which, basically withhold a letter. And this is where you get the reading frame change. So let's take a look at this. The normal one, you can see the original DNA, the resulting DNA, the resulting messenger RNA, and read the resulting peptide sequence. Methionine, lysine, leucine, isoleucine, stop. With a single insertion, you have now created a condition where you end up with methionine and then stop. With a single deletion, where you remove one of those X's, you end up with something really weird. You end up with methionine, asparagine, leucine, and tyrosine. Okay? So everything after methionine has been changed. Now, what are the effects? Well, a missense mutation, you have the original sequence of the codon changed due to the change in the nucleotide sequence. But then you get these other ones called frame shift mutations, and this is due to insertion or deletion of a nucleotide or nucleotides. The coding sequence of the gene is altered at the site of the insertion or deletion. The amino acid sequence changes from that point forward. If you see on text page 147, there's examples of that. You can have a spontaneous mutation, which is a naturally occurring mutation, occurs even though the cell is using an asexual means, such as binary fission, to reproduce. Hence, this adds to the genetic variation to the genome. But now we're going to move into something a little bit different, but much similar, and that is talk about mutations and mutagens and carcinogens. We've talked about mutations. They can be changes in the chromosome, but there are substances, chemical, physical, biological agents that increase the rate of mutation. The chemical agents are organic or inorganic chemicals. The physical agents, for example, are X-rays and ultraviolet light. Sometimes gamma rays also. The biological agents induce mutations through recombination, exchange of genes that leads to new genetic combinations. Now, we have a listing of carcinogens out there. These are mutagens that lead to the development of cancer. There's a whole host of this list. And in some places, California, for example, they have a higher standard, and this standard is held up by a piece of legislature called Prop 63. So if anything is used in that, basically, that product that looks like it would be in violation of Product 63, they have to post it on the item. Okay. Now, what about the Ames test? Because this is a test developed to determine if select compounds are mutagens. Now, there are methods that are used. Here's what they basically do. Ames developed this many years ago in the early 70s. He took a culture of Salmonella typhimurium and notice His negative, which means that it lost the capability to synthesize the amino acid histidine. Now, by exposure to a select compound, determine if the reversion mutations occurred. In other words, some of it was able to grow now on medium without adding histidine because it can make its own histidine. If you have this, then the compound that you put in there along with the Salmonella typhimurium is the compound is mutagen. So you have the first case of that. Now the Ames test was developed, but then people started saying, well, yeah, but some of the compounds that we take in that look safe, what if going through our biochemical process of digestion causes some chemical changes? Not all compounds are mutagenic at first, but they can be chemically altered by the organism, and an advanced Ames test was used to combine the subject compound mixed with the liver extract. What you do is you take your compound, you take a rat liver, you mince it up, mix it in with the compound, inject it all into a mouse, and then you would be able to see if the presence of the liver enzymes from the extract would help indicate that the original compound is safe, generally recognized as safe, or whether it is that some people now consider mixing it with such things as liver enzymes capable of creating mutations. Here we have our control, liver extract, medium lacking histidine, after incubation, 1, maybe 2. Baseline. Liver extract, suspended mutagen, put it down on here in this medium lacking histidine. And what do you have? 1, 2, 3, 4, 5, 6, 7, 8, 9, as opposed to 2. So the number of colonies is higher than expected for spontaneous mutation, 2 His plus. Next area we're going to talk about is DNA proofreading and repair mechanisms. These protect the stability of the genome. Errors in the replication of DNA can occur due to DNA polymerase. This is spontaneous and varies from organism to organism, yet the process can be corrected. We have various excision repair or error correction enzymes or clusters that are proteins that are based on, have sort of a mechanism, a process that basically will use specialized enzymes, and they'll clip out the damaged or mismatched nucleotides from the DNA. After the error is involved, polymerase 1 then replaces the correct nucleotide into the site. Then you want to seal off the ends. That's where DNA ligase comes in. Then seal the nicks, which they will, of the sugar phosphate backbone. Now there's another interesting one that pops up called thymine dimers. Now you think, I got to have x-rays. I got to have some strange chemicals that made the Joker go nuts. No, that's just fiction. Or maybe I got exposed to gamma rays. No, we won't go down that road and say you become the Hulk. But some cases, mutations can occur at a much lower intensity of electromagnetic radiation, in this case, ultraviolet light. If you have in your DNA 2 thymines next to each other, they can form what's called thymine dimers. This is the covalent bonding of neighboring thymine nucleotides on the DNA. And this occurs when UV light catalyzes the reaction. It can lead to DNA sequence errors. Because once you form these thymine dimers, running across it and getting the exact information is not possible. And so you have to have one, an excision complex that will detect it and then remove it. Two, DNA polymerase 1, which will help to take the break, refuse it together. And then finally, the entire break is brought together by DNA ligase. Next, what we're going to do is talk about some of the processes of gene transfer. This is extremely important because we're going to be talking about how you can basically have, let's say, a plasmid of disease resistance travel to other organisms and pass on these traits of antibiotic resistance. Now we have two types of gene transfer: vertical, horizontal. Vertical gene transfer is when the cells pass on genetic information to the next generation from parent to offspring. It can occur via asexual or sexual means. Horizontal gene transfer is the passage of genetic information between cells by a process independent of cell division, thus separate from reproduction. This can also occur across completely different phylogenetic domains. In other words, what about between this type of bacteria and that? What about between this type of bacteria and archaea? What about between this type of bacteria and euglena? Possible. Let's explore this. Plasmids, as I mentioned to you before, they're non-chromosomal DNA segments. They're found in bacteria and a variety of eukaryotic cells. They're usually circular. They commonly shared among cells by what is referred to as a horizontal transfer mechanism. Many can confer new traits, such as making toxins, protection against antibiotics, or the ability to perform conjugation. Now, the types of horizontal gene transfer we're going to talk about: conjugation, transformation, transduction, transposons. Okay? And the next thing someone might have asked was, well, what about transformers? No. This is not cartoonyville. Okay? Let's talk about conjugation first. This is the transfer of genetic material on what is referred to as a fertility plasmid. It's a particular plasmid that allows for exchange from one bacteria cell to another. You can't have it occur without the fertility plasmid, but this transfer will occur via a pilus, okay? Small segment of the cytoplasm and also the plasma membrane. This will form a bridge between two cells. Now, if the plasmid is completely transferred, the bacterium receiving the plasmid will have the trait of conjugation as well as be able to transfer other genes on the plasmid to other bacteria. And then we have what's called HFR, high frequency recombination. Now this is serious because this is where you have a plasmid that merges with the host bacteria chromosome. And the plasmid is no longer an independent plasmid of the cytoplasm. Yeah, okay, so what does that do? This is going to result in conjugation with other bacteria with the transference of parts of the bacterial chromosome along with the fertility factor. So what you have to think about is that this is a means in which you have recombination, but at a much grander scale. When I talk about recombination, this is an exchange of genetic material that will result in new genetic combinations.
We're going to have two bacteria, one here, this is resistant. Here's our antibiotic resistant gene. It's going to then make something called a pilus and transfer the gene over. Boom, it encodes this thing. Now we have an antibiotic resistant gene in here, and this organism now has the resistance to the antibiotics. Again and again, bacteria do this. Again and again, we see this happening in our bodies.
Moving ahead, That was an animation I wanted to give you as an example of this. Sometimes the animation makes it much more clear than just a static diagram. But to review, you have a plasmid that has the F factor, fertility. Here's your bacterial chromosome here, cell with F factor. Cell without F factor, this is their bacterial chromosome. In a very short period of time, the bacteria with the fertility plasmid builds a pilus, connects up with the neighboring cell, makes a copy of this plasmid with the F factor, and it will be then deposited into this cell. And now both contain the F plasmid. They both will now have the capability of genetic information exchange with other bacteria. Here's the HFR situation. Here is my chromosome, but the F factor integrated, merged into that bacterial chromosome. So all of this has the potential of being transferred over to a cell without the F plasmid. Here, I have a situation where I'm forming a pilus, and notice that part of the bacterial chromosome is going along in the copying, comes into this cell without the F plasmid. Now the F plasmid will be present in this new bacteria, as well as a piece of the HFR cell, and they will integrate into the regular chromosome of this cell. So it has the capability to have the HFR transfer. You picked up new traits coming from the primary bacteria. And so you're beginning to build, if we want to say it, a better bacteria, a more lethal bacteria. Here is a scanning electron micrograph of two bacteria with the pilus exchanging. Now, that's number one. Number two, transformation. Genetic alteration of bacteria once the bacteria take up DNA from their environment. Interestingly enough, we start finding out that bacteria cells have the capability to take pieces of DNA outside, floating around their environment, and take them up. So the transferring DNA that is taken up by that cell can be maintained by the cytoplasm as a plasmid or can be incorporated into the host cell's chromosome to result in a stably transformed cell. When we use the term competent cells, these are bacteria cells that can undergo transformation and express genes to allow uptake and processing DNA possible. Eukaryotes can undergo similar process of uptake of the DNA from the environment, but the term is used transfected, not transformation. Transformed in eukaryotes refers to abnormal cells with cancer-like features. Now, the first evidence of this was Frederick Griffith's experiments in the 1920s that provide evidence for this process. Basically, you had an organism, Streptococcus pneumoniae, with a capsule covering. This causes a lethal form of pneumonia. Without the capsule, no pneumonia. But if you killed the cells, no pneumonia. But if you mix heat-killed cells with living, non-encapsulated cells, you got a pneumonia. Huh? Turns out that the DNA, the genes to make the capsule that were in the heat-killed cells were taken up by the living, non-encapsulated bacteria, and they began to express the encapsulation genes. And they got transformed into bacteria that will cause pneumonia and can kill. So this was the evidence that DNA is genetic material, that environmental DNA can be transferred to other organisms and lead to new traits. This should be a helpful video.
One of the first steps toward the discovery that DNA is the hereditary material was made in 1928. A British bacteriologist named Fred Griffith was interested in two strains of the bacteria that caused the disease pneumonia. When viewed under the microscope, one strain of bacteria appeared rough. It was called strain R. The other strain had a mucus coat, and so it appeared smooth. It was called strain S. The two strains also differed in their effects when they were injected into mice. Mice injected with S bacteria developed pneumonia and quickly died. But mice injected with R bacteria did not develop pneumonia. They remained healthy despite the injection. Griffith wondered what could account for the difference in the ability of the two strains to cause pneumonia. He suspected the answer lay in the mucus coats, which were found only in the deadly S bacteria. Perhaps it was the coats that caused the disease. To test this hypothesis, Griffith heated the S strain bacteria over a flame. He reasoned that this would kill the bacteria without destroying their mucus coats. He then injected the heat-killed S cells into mice. Unexpectedly, the mice remained healthy. It appeared that the mucus coat did not cause the disease. Next, Griffith mixed together heat-killed S cells and live R bacteria. Since the S cells were dead and the R bacteria were harmless, he expected this mixture to be harmless. However, when the mixture was injected, it proved deadly. The mice developed pneumonia and died, just as they had when injected with live S bacteria. Griffith examined some blood from the dead mice under the microscope. To his surprise, he found live S bacteria. Where had these live S bacteria come from? Griffith concluded that when the bacteria were mixed, some material had passed from the dead S bacteria to the live R bacteria. This material had transformed the R bacteria into S bacteria. Most importantly, Griffith found that this change was inherited. When the S bacteria from the blood of the dead mice reproduced, they gave rise to more mucus-coated S bacteria, never to R bacteria. This led Griffith to conclude that the material that passed from the dead to the living bacteria must be the genetic material. Later experiments by other scientists showed that the transforming material was DNA. We now define transformation as the process whereby bacteria are changed by absorbing DNA from an outside source.
And you can see this scenario again here, aside of the video, where you take the mice, they're injected with living encapsulated S pneumonia, mouse dies. Non-encapsulated S pneumonia, mouse healthy. Heat-killed encapsulated S pneumonia, mouse healthy. Mix them, mouse dead. When they do autopsies, they examine the blood, etc. You have live encapsulated S pneumonia. The bacteria from the heat killed, because when the heat killed them, cells ruptured and they spilled out pieces of their DNA. The living non-encapsulated absorbed the DNA and thereby began to demonstrate the traits of that absorbed DNA. Finally, we're going to get into transduction. Now, this is the introduction of new genetic material into the bacteria cell by a virus. Bacteriophages, these are actually very specialized viruses that infect bacteria only. They're also referred to as phages. Once infected by the virus DNA, the phage DNA will be expressed and new viruses are constructed. Phage DNA will integrate, that is, combine with the host cell's chromosome. Now, generalized transduction, this is a random process, meaning whenever you take phage DNA, it contains some DNA from the previous host cell that was infected. The phage acts as a vector, a transfer vehicle for the host cell DNA. The new cell infected will have delivered and integrated into the chromosome the phage host cell DNA combination. As a result, the new bacteria will express new genes from the prior bacteria's DNA. Specialized transduction. This is a little bit different. It is the process where the bacteriophage packs specific bacterial genes into the new phage particle and then conveys those genes to the new bacterial host cells. So you're looking for specific characteristics. Now, temperate phages, such as the lambda virus, these are phages that combine with the host cell bacteria chromosome along with specific bacterial genes. They became sort of in the later 1980s the workhorse to do some of this genetic engineering. Now, upon a specific trigger, they emerge from the host chromosomes. They reactivate and leave the chromosome, build new viral particles with specific DNA host genes integrated into the viral DNA. Once the phage enters a new bacteria host cell, the new bacterial genes will enter, become active in the next host cell. So you're really looking at this. Generalized is more haphazard. Specialized is using unique phages with the idea of clipping out and creating, in essence, a library. Different bacteriophages have certain components of this bacterial DNA, and they will insert them into new bacteria with new combinations that eventually, hopefully, you will get what you're looking for on this piece of the DNA. This is a colorized scanning electron micrograph showing bacteriophages attacking E. coli. You can see them here, 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 etc. Okay, so remember that the virus, the phage, is much, much more smaller than a bacteria cell. Finally, we get into transposons, referred to by some as jumping genes. Now, in 1940s, work by Barbara McClintock described the transposable elements that cause various colors in the kernels of maize. You ever seen this? You have all sorts of different colors. Each kernel may have differing colors. Each kernel itself may have several different colors. What's a transposon? These are genes that shift their position in the chromosome, and in doing so, they may take along other components of that genome and put it in new places. So transposable elements are DNA sequences that move from one location on the genome to another. Now, there are two classes of transposons, retrotransposons and DNA transposons. Now, these processes can cause various human diseases as well as cause insertions and deletions in the genome. Retrotransposons, this is where DNA segments that get transcribed into RNA, then the RNA is reverse transcribed back into DNA, so we have a cDNA copy, via reverse transcriptase. The cDNA is then inserted into a new place on the genome. All insertions are risky, as they can interrupt or stop the normal DNA sequence, gene sequence. So think about it this way. Draw a big line, and then somewhere in there, just arbitrarily start dropping in a DNA with a different sketching of that DNA, and boom. What you may have had, if you want to use it from a CD analogy, CD music, okay? You're listening to a beautiful song, and suddenly it becomes an aria, and you're not really much of an opera fan. Or it could be a nice piece of country and western, and suddenly you get headbanger metal type of music. How did that happen? Something inserted itself into the code that was not desirable. But you have to also understand this. When you insert a certain amount of foreign DNA into a normal gene, one of two things can happen. One, you have an abnormal gene leading to illness or death or something. Or two, you end up with a much larger gene product that might be useless or might trigger somewhere in it a stop gene formed, and you end up with a truncated gene, either of which is not desirable for the normal physiological function of the cell. DNA transposons are referred to as mobilized DNA. There are several insertion strategies: cut and paste and copy and paste. Now, cut and paste, the transposon DNA cuts out of the DNA and inserts into a new DNA site. This results in a gene interrupted. The copy and paste, the DNA with the transposon copies the DNA section, then excises itself, pulls itself out, and inserts into a new site, usually interrupting another gene. The way to think about it here, here's retrotransposons. I've got my DNA, I made it into RNA, I made a copy into cDNA, and now it gets acted and inserted somewhere else. This is what I meant by the various kernels of maize. Notice some are black, some are orange, some are yellow, etc. And some actually have multiple colors in them. Copy and paste mechanism, I take the DNA transposon. What I have are these couple of little segments here at the edge that allow it to lift itself out and then insert somewhere else. This targeted gene may be interrupted or shut down in its functionality. Copy and paste, what happens is I literally pull this gene out. And now I've got a gap left in the DNA, and it inserts itself later on in here. Again, this targeted gene is interrupted by the DNA transposon. Now, to help you, this is a nice little summary of it's trans something. Transcription, translation, I taught about that, okay? To transcribe is to make a copy. To translate is to go from one language to another. In this case, the language of nucleic acids down to the language of amino acids. Transformation, you're transforming when you pick up DNA from the environment. Transduction is when bacteriophages steal pieces of the DNA in the cells and then shuttle them into the DNA of another cell. Transposon, this is where you can literally take pieces of DNA, jump over to somewhere else on the chromosome, or onto another chromosome entirely. Well, that's it for the genetics right now. I want you to keep in mind that some of this is going to be very applicable later on, particularly when we talk about the various states of horizontal gene transfer. So you may want to hot page this area, okay? Because we'll come back to it, particularly when we talk about antibiotic resistance and other things later on. I do encourage you to review the visual summary, which is on pages 158, 59. Review the chapter overview, which is on page 160. Review the end of chapter questions, which are on page 162, 163. Also, don't forget that you have work in Mastering Microbiology. Homework, which is due Friday by midnight. Quiz, which is due by this Saturday, midnight. Do take the time to review the videos in Blackboard for lecture content. Next up, we're going to be going over viruses and prions. That's chapter 6. And I hope you all have a nice day.